today we're introducing you to a gene editing technology called CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it's mediated by the Cas9 enzyme. The development of this technology is extremely recent, and by recent we mean it was first shown to work in humans just 3 years ago in 2012. So human genome engineering technologies like CRISPR can be exciting, scary, and controversial. And this is because they invite us to ask really big questions about our future as a species. First of all, will we be able to find new treatments for life-threatening genetic disorders that are currently incurable? And then on the flip side, will we abuse this power to develop superhuman traits? And so we're going to start this emotional discussion where many other emotional discussions have begun with Brad Pitt and Angelina Jolie. So Angelina Jolie has a mutation in the BRCA1 gene. And this mutation, the BRCA1 gene is a tumor suppressor. And so having this mutation makes her significantly more likely to develop breast and ovarian cancer. As a parent, she is also likely to pass this genetic predisposition on to her children. So what are Angelina's options if they want to have kids, or other parent options if they're there? Their first option is one, have kids, who are likely to inherit that mutation. And then secondly, to adopt or not have kids. But what if there was another option? What if there was a way to test an embryo for unfavorable genes, identify those genes, and then replace or edit those genes with better genes? This brings us to CRISPR. Scientists discovered CRISPR as part of the prokaryotic immune system. And what CRISPR essentially does is it uses the Cas enzyme to recognize specific foreign DNA and then cut that double-stranded DNA at specific sites. The key word is specific. So the Cas is able to specifically recognize the site on the DNA, which leads to cell death in the system. It was only recently that scientists realized that they could take CRISPR and use it in gene editing processes across organisms. So in this process, we see the same thing happening. Cas9 again acts as a restriction digest enzyme, guided by an RNA guide sequence, and then the targeted gene is cleaved out. A DNA template with the desired gene is then added to the cell, and the cell machinery repairs the DNA based on this template. So while CRISPR has been developed in a variety of organisms, including crops especially, some of the most exciting developments have occurred in monkeys. And this is because monkeys can readily serve as models for a variety of human diseases. So far, scientists have successfully engineered monkeys with targeted mutations in genes involved with regulating metabolism and improving immune function. However, their experiments were unreliable, with a low success rate of only about 10 to 25%. When it comes to CRISPR developments in humans now, we need to differentiate between somatic and germline treatments, as the last group did. Companies like Editas Medicine, which is based right in Cambridge, so right next door to us, are trying to use CRISPR to treat diseases in somatic cells. The main challenge with this technology is how do you deliver this technology to all of the cells in the region of the body that you want to treat. Nevertheless, gene editing technology for somatic cells have the potential to treat a number of diseases. First, we can see CRISPR potentially being used to treat cancer by inducing tumor suppressors at the site of the cancer. Secondly, CRISPR could also be used to treat sickle cell anemia by modifying DNA within the enhancer region of the sickle cell gene in order to circumvent the defect. And then finally, we could see it treating HIV through interrupting the HIV-1 provider. So germline treatments in humans are where most of the controversies lie regarding the use of genetic engineering ethically. This year, in 2015, a Chinese team edited 80 human embryos using CRISPR, and a handful were successful. The main problems with this were unexpected mutations that developed, mosaic, and how some embryos ignored the DNA template and repaired themselves using the original gene, which means that it did not make anything happen. Now, while there is tremendous excitement about the potential of this technology to cure genetic disorders that are life-threatening, the unintended side effect is that they also give us the ability to alter other traits, which raises really open-ended questions about our future. Should we choose how we look? How smart we are? What sort of body type we're likely to have? And the biggest question of all is, where will this technology stop? There are also a few other concerns ethically that we need to weigh in thinking about this technology. First is the number of monkeys and human embryos that we will need to test on in order to develop this technology. Secondly, there's also the problem with sickle cell anemia, where an unfavorable trait has an unexpected evolutionary advantage. By eliminating mutations in our genome, are we hurting the diversity in our species and thus hurting us as a whole? There There are also some legal concerns that we need to take into account. So this map shows in red all of the places where there are bans on using CRISPR in humans. The gray countries are where there are significant restrictions in place. As you can see, the most important thing to see from this map is that there is not a global consensus. So there are some countries that are developing this technology very quickly, while others cannot develop it at all. So in order to reach this sort of international consensus that we need about this technology, the thing we need most is dialogue. And right now we can see dialogue happening in a variety of ways. George Church, a geneticist at Harvard Medical School, has called for a moratorium on using CRISPR technology in humans until we've proven that it's safe and ethical standards have been made for using it. At Johns Hopkins, an international team of experts has been assembled to basically develop these policies for utilizing this technology. So finally, bringing this discussion all the way back to Brad and Angie, we can see both the potentials and problems with this technology. If it really can fix predispositions to diseases and cancers like the BRCA1 mutation, this could completely revolutionize medicine. However, if this technology is only made available to the rich and elite, it could further divide our society and our world.
Yay. So, based on both scientific and ethical considerations regarding human genome engineering, here are our propositions. The first is that